Good morning. Yesterday, we started something and also we completed regarding to the small structure, whatever we need of DNA. Then today we should be go to continue for the remaining what you need regarding to the structure of DNA. So, means in between two helicals, the 20 angstroms is nothing but the 2 microns. The diameter we can say. And the chemical composition of DNA, the nitrogen bases, sugar and phosphate. Then nitrogen bases, two groups, purines and pyrimidines. Purines are adenine, guanine, cytosine, thioine. So, in the structure of these two benzene cycles are there. In these, a single benzene cycle is there. So, due to the repulsion in between same poles, no bond located in between AG. And where the bond takes place in between opposite poles means a purine bonds with pyrimidine. Then the pyrimidine is bonds with purine. Then purine bonds with pyrimidine. Like this. The alternate, the bond is located. Then what the bond is located in between nitrogen bases means A bonds with T, C bonds with G. So in between AT double bond and CG triple bonds are there. The both sides the nitrogen bases binded with sugar. Then both sugars binds with phosphate. A bond is phosphodiester. Phosphodiester bond located. Then this side also the sugars are there, both sugars combined with phosphate. For split to bond, a hydrogen bond in between DNA, the required enzyme is helicase. For split to the glucoside bond, which takes place in between nitrogen base and sugar, the glucosidase enzyme is required. For split to phosphodiester bond, they required an enzyme, chlorophyllase. Yesterday I given all these things. Then what concern? In DNA, the coils are there, loops. If you take any loop, the core portion is bonds with the nitrogen bases. A bonds with T is in core portion, in the middle of the coil. Then both sides, sugars are there. A, T, C, G. Then sugars combined with phosphate. So entire the structure of DNA depends on phosphate only. If we are removing phosphate, what happens? The structure of DNA becomes to collapse. So entire DNA structure depends on phosphate. Why? Because it is the base number you see. Sugars are 2, nitrogen bases are 2, but the phosphate group is single. It is the base number. So the structure of DNA depends on base number. And also A, T, C, G, G, C is there. C, G is there like this. If you take any, how many pairs if you take whatever, what concern here, the number of phosphates are low from compare with the number of sugars and nitrogen bases. How many nitrogen bases means 4. Here also 4 sugars, but phosphates are 3. So if you are removing simply the phosphate group from the DNA, what happened? The structure of DNA becomes to collapse. Means the phosphate group is a backbone. What is the backbone for the DNA means? The DNA structure depends on phosphate group. So phosphate group is known as backbone of DNA. What is the backbone of DNA means? A phosphate group is a backbone of DNA. Is it clear? Just we should go to just little bit of the moving further forward. So here you see it is the nitrogen based sugar. Nitrogen based sugar, nitrogen based sugar, nitrogen based sugar. How many nitrogen bases are there? 4 means adenosine bonds with sugar. Guanine is bonds with sugar. Cytosine is bonds with sugar. And thymine is bonds with sugar. What is the basic concept here? Means the nitrogen base, including sugar, collectively known as glucose, means nucleoside. What is the nucleoside? Nucleoside means a combination of nitrogen base with sugar. A combination of nitrogen base with sugar, known as nucleoside. What is the nucleoside means? Nucleoside is nothing but the combination of nitrogen base with sugar. So how many nucleosides are there? 4. How many nucleosides? 1, 2, 3, 4. These are the 4 nitrogen bases with sugars. So the total number of nucleosides in DNA, if you take how much length of the DNA, even they are made up with the 4 types of nucleosides. The total number of nucleosides are 4. Then one more point here. Nitrogen base, sugar, again phosphate. You see, both. This phosphate is concerned to this nucleoside and this phosphate is concerned to this nucleoside. How many nucleosides are there? 4. Then what about the phosphate if is including with nucleoside, collectively known as nucleotide. What is a nucleotide? What is nucleoside means? Nucleoside, nucleotide. Nucleoside means nitrogen base with sugar. Nucleotide means nitrogen base sugar with phosphate. The total number of nitrogen bases, the total number of nucleosides are equal for the total number of nucleotides. How many nucleosides? Adenosine bonds with sugar, thymine bonds with sugar, and guanine bonds with sugar, cytosine bonds with sugar. These 4 collectively known as nucleosides. In other hand, nitrogen base, adenosine, sugar plus phosphate, thymine, sugar with phosphate, and guanine, sugar with phosphate, and cytosine, sugar with phosphate, collectively known as nucleotides. Nucleoside, nucleotide. The total number of nucleosides are equal for the total number of nucleotides. Means what I can say, in the coil of DNA is filled with nucleotides like this. If you take a coil of the DNA, if you take any coil, is filled with nucleotides. How many nucleotides enclosed in a coil is 20. This coil is filled with 20 nucleotides. And this coil is filled with 20 nucleotides. If you take any coil of the DNA, is filled with 20 nucleotides. Is it clearer? How many nucleotides? 4. How many nucleosides? 4. The coil of the DNA filled with how many nucleotides? 20. You see, the nucleotides are arranged in always in pairy nature. Always pairs. How many nucleotides? 4. How many nucleosides? 4. What is the backbone of DNA? Phosphate. Due to the base number, what we concerns to the structure of DNA is depends on phosphate group. Just simply, if you are removing phosphate group from the DNA, the entire structure of DNA becomes to collapse. Why? Because depends on phosphate structure. Then a coils. Yesterday I given the coil means due to the attraction in between opposite poles, what happens in clockwise, the two helicals of DNA becomes to spiraling, a coiling. They forms two coils, and each coil is filled with how many nucleotides? 20, and nucleotides are always arranged in the pairy nature. So each and every coil is filled with 20 nucleotides, or we can say for the 10 pairs of the nucleotides. The number of nucleotides is 20. But how many pairs? 20 means 10 pairs. Means the coil is filled with 20 nucleotides or 10 pairs of nucleotides. Then what is the conclusion today? Means yesterday filled with 20 nucleotides or 10 pairs of nucleotides. How many? What is nucleotide means? Nitrogen base, sugar, phosphate. How many nitrogen bases? Adenosine, sugar, phosphate. Guanine, sugar, phosphate. Cytosine, sugar, phosphate. Thiamine, sugar, phosphate. These four nucleotides. Repeatedly, again and again. Alternating. They can be arranged in coils of the DNA. And always the nucleotides arranged in pairy nature. And each and every coil of the DNA is filled with 20 nucleotides. The 20 nucleotides is nothing but 10 pairs. Today, what we are taking the clarity regarding the DNA structure means a coil filled with 20 nucleotides or 10 pairs. Nucleotide in the sense what we can say the nitrogen base, sugar and phosphate. And the total number of nucleotides are 4. And total number of nucleosides are 4. Nucleoside in the sense the nitrogen base, if it is combined with sugar only, we can say as the nucleoside. The total number of nucleosides are 4. The number of nucleosides are equal for the number of nucleotides. Is it clear? Okay. Next, one more point you will see. Then DNA is there. It is filled with what can be located in coil? How many nucleotides? Yes. Then you see, but what is the concept here means the DNA is made up with genes. What is meant by gene means a smallest micro unit of DNA, a undividable micro unit of DNA. Undividable micro unit of DNA is known as gene. But basically what we are concerned here means the DNA is made up with nucleotides. Nucleotides enclosed in coil. In other hand, we can say as the gene. What is gene means? A gene filled with them. Coils. And coil filled with the nucleotides. And basically gene made up with nucleotides. How much part of DNA is considered as the gene? How much part of DNA is considered as gene? Micro means how much? How much micro? No, what I can say, what the part of DNA is considered as the gene means, the gene is defined as a smallest part of DNA which determines to a character. Each and every character of body has been going to controlling by a genes which are located on DNA. You mind this point, what I can say? A smallest part of the DNA which determines to a character. A smallest part of DNA which determines to a character, single character. A part of DNA. A part of DNA which determines to a character. The part is known as gene. For example, you see the skin color is there, determining by a gene. Age color is there, determining by a gene. Visibility is there, determining by gene. Memory power is there, determining by gene. A secretion of insulin, determining by gene. All the hormones are secreted from genes. What I can say, gene means a smallest DNA which have the capacity to proliferate to a single character. The part of DNA is considered as gene. Is it clear or not? What I can say? A part of DNA which determines to a character, the part of DNA is considered as gene. Gene in the sense, in Telugu we can say as genuvu. What is genuvu? What is gene means? A part of DNA. What is part means? A smallest part. How much smallest part means? A character proliferating. A character determining. A smallest part of DNA is considered as gene. How much smallest part of DNA is considered as gene means was later they was calculated. Means basically you see a DNA, a coil filled with nucleotides are not, 20 nucleotides or 10 pairs are there. But what I can say here, the part of DNA which have the capacity for proliferate or determines to a character, the part is known as gene. And basically gene made up with coils. And a coil made up with nucleotides. So basically gene made up with, nucleotides. How many nucleotides? How many nucleotides they required for the proliferating to a character? Basically, a gene means a minimum number of nucleotides, how much they required for proliferating, determining to a character means was calculated. 900 nucleotides part is required. So where the gene, if it is filled with 900 nucleotides part of the DNA, have the capacity for proliferate, for determines to a character. How many? 900 nucleotides part of DNA have the capacity for proliferate to, for a determine, for a differentiate to a character. Means 900 nucleotides arranged in how many pairs? 450. A coil filled with how many pairs? 10 pairs. Coil with 10 pairs. Total number of nucleotides? 900. Minimum number of nucleotides, how much they required for a differentiate, a determines to a single character, means a 900 nucleotides part of DNA determines to character. Maximum number of nucleotides they required for the 1500. The minimum number of nucleotides, how much we required for determines to a character by a part of DNA is nothing but the 900 is required. Maximum number of nucleotides, how much they required means 1500. Means 900 to 1500 nucleotides part. Also known as 450 pairs to 750 pairs of the DNA part. And the pairs enclosed within the coil by 10 means 45 coils to by 10 means 75 coils part. Understood? What I can say, a DNA differentiated into genes, a DNA filled with genes. How much part of DNA is considered as the gene means minimum number of nucleotides when they are enclosed within the DNA 900 to 1500 nucleotides, or even we can consider the 450 to 750 pairs of nucleotides part, and also a 45 coils part of DNA or 75 coils part of DNA have determines to character. So this portion considered as gene. Is it clear? So what is meant by gene means a smallest part, a undividable, a smallest part of DNA is considered as gene. Undividable, undividable, smallest part of DNA is considered as the gene, and gene made up with nucleotides. Then how many nucleotides enclosed in a gene means in between 900 to 1500 nucleotides. So 900 means always the nucleotides are arranged in pairy nature. In between a 450 to a 750 pairs of nucleotides part is also considered as the gene. And then pairs of nucleotides enclosed in a coil enclosed in 10 pairs means a 45 to 75 coils portion have the capacity for proliferate, for differentiate, for determinate to a character. The part of DNA is considered a gene. One more definition also there. - 900 to 1500. Then this point, what is this point means later we go to discuss. Not itself. A centromere we can say is the different. But we have the two helicals are there. The coil filled with the nucleotides. Once again, again and again I am going to explain why, because for getting somewhat what you need. Then 9 nucleotides, pairs of nucleotides, coils is equal to gene. What is gene? A determining factor of DNA. What the determining? A character determining factor of DNA. How much part of DNA determines to character? Means 45 a coils portion or 75 a coils portion of DNA have capacity for determines to a character. The determining portion is known as gene. The name gene was given by Johansson, not Mendel. What is gene means? The name gene was given by Johansson. What is gene? What is gene means? A character determining part of DNA is known as gene. And also gene considered as from parents to offsprings. A character, if it is carrying by a part of DNA, how much part of DNA is carrying to a character from parents to next generation? The part of DNA is also known as gene. Then parents are there. For example, a character, a transfer to next generation by a smallest portion of DNA is considered as the gene. The transfer of character from parents to next offsprings is known as heredity. What is meant by heredity? What is meant by heredity? A transfer of characters from parents to next generation, next offsprings, next progeny. The character What can be the characters of the parents can be ultimate transfer to the offsprings or the progeny. How they can transfer means in parents the gene is there. A gene is the fundamental unit for the heredity. Through a character, through a gene, the character can be transferred to next progeny. Means what I can say, the part of DNA which have the capacity for transfer to characters from parents to next progeny, the part of DNA is considered as gene. What is the a modern definition for the gene means a gene is a smallest part, a undivided portion of the DNA is known as gene. Then how much portion of the DNA is considered the gene means in between 45 to 75. A coil's portion of the DNA is considered the gene. A hereditary unity, a fundamental unit of heredity. What is the fundamental unit of heredity means? The characters whichever are transferred from parents to next progeny is known as heredity. The heredity is transferred through a gene, a smallest functional, a part of the DNA which have the capacity but transfer to character from parents to next progeny. The part of DNA is considered also gene. So gene is a smallest unit for a heredity. Smallest unit, undividable smallest unit of the heredity is also considered as the gene. Then also a modern definition in Telugu Academy they given. What we can concern here you see during the time of protein synthesis. Cell, a unit of the metabolism. Whatever the metabolism is located in our body, the smallest unit is the cell. Then cell, what happened here, they are synthesizing to proteins on mRNA, something just you go to listen. You won't felt in confused. Just what I am giving, just for a little bit of either 1 or 2 points we need. But that only we are discussing here only. What I can say, in cytoplasm, the mRNA and ribosomes, they can be synthesized to proteins. And what is the basic concept mean? The protein is made with amino acids. Then amino acid is secretes from a smallest part of DNA. You see. A gene have the capacity for release to amino acids. These amino acids can be formed in the polypeptide chain. Then the polypeptide chain forms to protein. Means during the time of protein synthesis, the polypeptide chain prolongating part of DNA is also known as gene. For a gene, we have the either 3 or 4 definitions are there. What is gene? In between 900 to 1500 nucleotides portion of DNA is considered gene. Or 750, I think the 450 to 750, if this nucleotides portion of DNA is known as gene. 45 to 75 coils portion of DNA is known as gene. A character whichever transfer from parents to next progeny, the part of DNA is also considered as the gene. In the definition for the moderation, what concerns to the gene means during the time of protein synthesis, the polypeptide chain prolongating part of DNA. Polypeptide chain prolongating means they can be synthesized to amino acids, the enclosing within the polypeptide chain finally leads to the formation of protein. The protein formation determining by the polypeptide chain, the polypeptide chain amino acids can be secreted from the part of DNA. Means what I can say during the time of protein synthesis, the part of DNA which have the capacity for proliferate to a polypeptide chain. That much of a portion of the DNA is also considered as the gene. For gene itself, we have the 4 definitions. They may be give a question for the 1 mark question, or there may be choices in preliminary. And also we need a more explanation for right to generalizing. Also need for the biotechnology itself. Also need its need for RDNA technology. Before enter to the RDNA technology, we should get the basic knowledge and basic points what we need. Then only we are trying to get the clarity from these aspects. Okay. Then here what I can say in DNA. Then today what we discussed here means in DNA, just we have the nucleosides are there and nucleotides are there. Gene. What is meant by gene means a smallest unit of heredity. A part of the DNA which made up within between 45 to 75 coils portion of the DNA. Again, gene is defined as the during the time of protein synthesis, the polypeptides and prolongating part of DNA is known as gene. Is it clearer? Now you go to take notes, please. Yesterday for the continuation. What is the last point yesterday given? No. Yes. DNA. DNA made up with nucleotides. DNA made up with nucleotides. Nucleotides. Yes, you make the side heading for the nucleotide. What is nucleotide? You make the side heading. DNA made up with nucleotides. You make side heading for the nucleotide. Nitrogen base, sugar with phosphate. 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 Known as nucleotide. Known as nucleotide. These are four. ఇది ఒక్కటి రాయి. Okay. These were arranged in coils. These were arranged in coils. In fairy nature. In fairy nature. These were arranged in coil. In fairy nature. These were arranged in coils in fairy nature. Each coil filled with 10 pairs. Each coil filled with 10 pairs are 20 nucleotides. Each coil filled with 10 pairs are 20 nucleotides. 20 nucleotides. 20 nucleotides. One more point here you see. ఒక చిన్న సవరణ. ఈ పేపర్ సిలబస్ అయిపోయేంత వరకు కంటిన్యూషన్ ఉండాలి, ఆబ్సెంట్ ఉండదు. ఇంకొక ప్రిన్సిపల్ ఏంటంటే లేట్ రావద్దు. ఈ లేట్ రావడం వల్ల కూడా ఉపయోగం. 10 minutes లేట్ వచ్చినా 2 minutes లేట్ వచ్చినా అది పోతుంది. అంటే ఆర్డర్ తప్పుద్ది. ఆర్డర్ తప్పితే మీ ఉపయోగం ఏం లేదు. కాబట్టి ఫ్రాక్షనల్ టైం అన్నమాట. టైంకు వచ్చేటట్టు ఉండాలి. 2 నిమిషాల ముందు ఇక్కడ ఇది కూడా అట్టే ఉంటుంది అయిపోయేంత వరకు. ఈ తర్వాత మీరు చదువుకోవచ్చు, సపోర్ట్ చేయాలి. సరే కదా. దెన్ how many pairs? 10 pairs అన్నాం. A, T, C, G, G, C, T, A. Like Smith. How many pairs here in a coil? 10 pairs. And one more point here, the orderly nucleotide, it is the nucleotide, it is nucleotide, 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 like this. These are the pair. These are pair. Wilkins, who was calculated, who? Wilkins was calculated. Orderly, any two nucleotides in a coil can be arranged in the length of 3.4 angstroms. Today, we go to discuss to the microstructure of DNA only. If not easy to us, it may be complicated for the biotechnology, raw DNA technology. అది లేవాలంటే ముందు తెలవాలి. దీంట్లో 3 ఆప్షన్స్ ఉన్నాయి. 2 మార్క్స్, 5 మార్క్స్, 10 మార్క్స్. లేదు పాడుకూడదు, ప్రిలిమినరీలో చాలా ఇంపార్టెంట్ ఆస్పెక్ట్ కాబట్టి దీన్ని ఏం చెప్తున్నానో దాన్ని చదివేశాను. కష్టం క్లిష్టం అనుకోవద్దు, పర్ఫెక్ట్గా చదవాలి. ఎస్, నేనేమంటున్నాను, ఆర్డర్లీ 2 nucleotides which are arranged in pair in nature, Wilkins was explained and also was stated. Any orderly 2 nucleotides, the length is 3.4 angstroms. A coil filled with how many pairs? How many pairs? 10 pairs. In a pair, what is the length? 3.4 angstroms. In a pair, in between these 2 nucleotides, orderly 3 arranged for 3.4 angstroms was explained by Wilkins. How many pairs in a coil? In a pair, what is the length? 3.4 angstroms. The total length of coil, what is the total length of coil? 10 into 3.4 angstroms is equal to 34 angstroms. Was explained by Wilkins. Simply we can extract. What I can say, Wilkins was explained, the nucleotides can be arranged in a coil. A pair of nucleotides orderly, they can be arranged in the length 3.4 angstroms was calculated. And also was stated, a coil is filled with 20 nucleotides or 10 pairs. In between a pair 3.4 angstroms. And also was stated the total length of coil is 34 angstroms. Then, in a coil, how many pairs? 10 pairs. You see, what is the angle in between these 2 orderly nucleotides? What is the angle? How many degrees angle? It is the coil. AT, CG, GC, TA, AT, like this. Sugars are there. Both sugars, phosphate. Orderly, in between these 2, 3.4 angstroms. Then the total length of coil is 34 angstroms. Then what about the how many degrees angle in between these 2 nucleotides means? It is circular in condition. A coil, what is the shape of coil? Circle. Circle is equal 360 degrees. Then how many pairs in a circle? 10 pairs. Means, yes, the 36 degrees angle arranged in between any 2 orderly nucleotides of DNA even also was explained by Wilkins. How many degrees? 36. What is the length of coil? Length of coil? 34 angstroms. Length in between 2 nucleotides? 3 point. In between 2 helicals? 20 angstroms. In between 2 helicals, 20 angstroms. In between 2 nucleotides, 3.4 angstroms. The length of coil is 34 angstroms. Then in between any orderly 2 nucleotides of the coil, they can be arranged in 36 degrees angle. How we can say the coil is in circle condition? The circle is 360 degrees, filled with 10 pairs. If you want a pair, 36 by 10. Sorry, 360 by 10. Then we can get the 36 degrees angle. Is it clearer? Then, ఎక్కడ ఆగినాను చెప్పండి. అమ్మా అని చెప్పండి ఒక్కరే. Wilkins was explained. Wilkins was explained orderly, a pair of nucleotides, a pair of nucleotides arranged in 3.4 angstroms length, and coil filled with 10 pairs. So he was estimated. The total length of coil, 3.4 angstroms into 10 pairs. 3.4 angstroms into 10 pairs is equal to 34 angstroms. And also he was stated a coil is in circle shape equal for 360 degrees so in between a pair of coil, 360 by 10 is equal 36 degrees. Okay, next you see adenine, guanine, cytosine, thymine. But what is the nature of bond? Where it is bond is located in between, not in between AG. The bond is in between AT only, due to the opposite poles, not in between AG. So A bonds with T. And C bonds with G. Then what is the shape in between these 4 nitrogen bases? Square shape. Is it not square? Means nitrogen bases are arranged in square shape. In a square, in each angle is filled with 90 degrees. Here also 90, here also 90, here also 90. What was I given instruction for you? Either the 25% of the matter what we are preparing in the notes, if we are omitting, even it is equal for 100%. Don't bother about the application. So, just we are taking a little bit of a small an account regarding. So, what I can say, if you take a DNA structure, adenosine bonds with thymine, cytosine bonds with guanine, and these 4 arranged in square shape. What I can say, in between A and T, how many degrees? 90 degrees. If you take any 2 of nitrogen bases, they are arranged in 90 degrees. AC even 90 degrees. And CG even 90 degrees. And GT even 90 degrees. If you take any 2, why? Because the nitrogen bases are arranged in square shape. So in between any 2 nitrogen bases, the angle is 90 degrees. Is it not correct? Note this point. Nitrogen bases. Nitrogen bases arranged in nitrogen bases arranged in square shape. Nitrogen bases arranged in square shape. So any two nitrogen bases, 90 degrees angle is present. 90 degrees angle is present. Just you take this rough diagram regarding this one, enough. You see here A, T, C, G. Sugar, phosphate. What are these two? Purines. A plus G, purines. What are these two? S. How many degrees here? Here also A plus G is equal to how many degrees? 90 plus 90. 180. Similar here also 90 means the ratio in between purines and pyrimids of DNA is 1:1 was explained by Chargaff. Chargaff, a scientist, who was explained. What he was explained means the ratio in between purines and pyrimidines of DNA is 1:1. The ratio in between purines and pyrimidines of DNA is 1:1. How we can prove means like this. 1:1 means the ratio of purines and in pyrimidines is 1:1. Explained by Chargaff. So the Chargaff law. What is the Chargaff law means? The ratio in between purines and pyrimidines of DNA is 1:1. It was explained by Chargaff, so known as Chargaff law. Not this point. In DNA, angle in between purines, is 180 degrees. Angle in between purines, 180 degrees. And in between pyrimidines, 180 degrees. 180 degrees. So the ratio in between purines and pyrimidines, is 1:1. 1:1. Known as Chargaff law. Next, one more point. A DNA filled with genes. DNA filled with genes. DNA filled with genes. Yes, you make the side heading for gene. What is meant by gene? You make the side heading. Gene means gene. It is the smallest undividable unit of heredity. The smallest undividable unit of heredity. The smallest undividable unit of heredity made up with in between 900 to 1500, made up with in between 900 to 1500 nucleotides, or 450 to 950 pairs of nucleotides, 450 to 750 pairs of nucleotides, or 45 to 75 a coil's portion of DNA. A coil's portion of DNA. 45 to 75 coil's portion of DNA considered as gene. Considered as gene. Modern definition and pattern. Modern definition. Modern definition for gene. Again, you make the side heading. Modern definition. Modern definition. Modern definition. What is the modern definition means? During the time of protein synthesis. During the time of protein synthesis. During the time of protein synthesis. A part of DNA. During the time of protein synthesis, a part of DNA. Protein synthesis. During the time of protein synthesis, a part of DNA which prolongate a part of DNA which prolongate to polypeptide chain. Polypeptide chain. A prolongate to polypeptide chain. A part of DNA which prolongate to polypeptide chain. Prolongate to polypeptide chain. Polypeptide chain. The part of DNA is known as gene. Part of DNA is known as gene. కొంచెం ఒక్క నిమిషం కింద. This portion prolongate to polypeptide chain. So this portion is known as gene. And DNA where the gene arranged, the place is known as place. Where gene arranged on DNA. This, how much length? This much of place. కొంచెం వినాలి. It is a gene, maybe 45 coils are there. Proliferate to polypeptide chain. This part of DNA is considered as gene. But the gene arranged on DNA, where it is in a place. The arranging gene arranged in a place on DNA, how much place they required for the arranging itself? The place is known as locus. What I can say? The locus means the place where the gene arranged on DNA. Place, particularly place they required for the arranging itself. How much place they required? The place is known as locus. Is it not clear? What is the locus means? A place where the gene is arranged. Where gene arranged on DNA. The place is known as locus. Means one locus is equal to one gene. Gene means a particular portion. For arranging to gene on DNA, they require a place, a small portion. The place of DNA where the gene arranged The place is known as locus, and locus filled with a gene. Locus filled with gene. But generally, 99% the locus filled with gene. Sometimes we can say, a recent views what they are explaining, a locus filled with more than one genes. Means multiple genes can be arranged in the same locus. What is locus mean? The place where the gene arranged. What is locus? Place where the gene arranged, the place is known as locus. Generally, the locus filled with a gene. But recent views, what they are explaining means a locus is filled with more than or the multiple genes. At the time, the locus is known as loci. What is loci? What is loci means? The plural number for the locus. A singular number and plural number. The place where DNA, the place where the gene arranged on DNA, the place of DNA is considered as the locus. Generally, locus filled with the gene. But if the more than one genes, if they are arranged in the same locus, at the time the locus is called as loci. The loci is the plural number for the locus. Just you make it. Genes arranged. Genes arranged. And locus. Genes arranged on locus. The place where gene arranged on DNA, the place where gene arranged on DNA, place where the gene arranged on DNA, the place is known as locus. The place is known as locus. Locus filled with a gene. Locus filled with a gene. Locus filled with a gene. Sometimes locus filled with more than one genes. Locus filled with multiple genes or more than one genes. Locus filled with multiple genes or more than one genes. At the time, locus called as loci. Locus called as loci. 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 It is a plural number for locus. 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 Plural number for locus. Next you see now what is the smallest portion of DNA is gene. What is the function of gene means? What is the function of gene? The secrets to amino acid is మర్చిపోదు. A fundamental, a basic function. They can be secrets to amino acids. Later in cell, the group of amino acids they are able to form for the protein. Means amino acid turned into protein. Then based on this character and structure, protein can be divided into enzyme, hormone, or pigments, or poisons. What is poison? Yeah. Basically, all the poisons are basically proteins. Saliva. What is saliva? Saliva. Salivary gland secretes to saliva. Saliva secretes from salivary glands. Where from the salivary glands means in salivary glands, in cell, in nucleus, in chromosome and DNA, gene is the responsible. It can be secreted to a group of amino acids that turned into a protein, then protein can be turned into saliva. Means basically the saliva is a protein. Saliva of snake, is it not protein? Is it not protein? A snake bite, if it is injecting to the saliva into our body, is it saliva to ourselves? A poison. What is the poison means? Antigen. What is the antigen means? A foreign particle of our body. A foreign particle, if cannot be sustaining in our body, is considered as the poison. It is nothing but antigen. What is antigen means? Antigen is a poison. A poison is basically made of the protein, made of with amino acids and secretes from a gene. ఇక్కడ మనం ఏమంటున్నాం? A fundamental function of the gene is the secretion of the amino acids can be turned into protein. Then protein, that structure and character, function, they can be differentiated into enzymes, hormones, pigments, proteins, and antibiotics. Means all the antibiotics are proteins. If not sustainability to our body, it is a poison to us. The fundamental function of gene, fundamental function of gene is secretion of fundamental function of gene is secretion of amino acids. Then it forms two proteins. Based on structure and character. Protein turned into structure and character. Proteins turned into enzymes, hormones, pigments, enzymes, hormones, pigments, poisonous substance. ఏమన్నావు? Poisonous substrates and also antibiotics. Lipids. నేను చెప్పింది మర్చిపోలేదన్నమాట. లిపిడ్స్ రాయవచ్చు. రైట్. ఓకే. A fundamental function of the gene is the secretion of amino acid, can be forms to protein, then proteins based on their properties, structure and character, they can be turned, molded as an enzymes, hormones, pigments, poisonous substrates or antibiotics. ఇవన్నీ కూడా బేసికల్ గా జీన్ సెక్రెషన్సే. ప్లీజ్ ఇంకొక నిమిషం. What is the character of DNA means? Replication, duplication, autocatalysis, Xerox. A terrific character. You see, two helicals are there, 3 dash, 5 dash, 5 dash, 3 dash. They can be able to coil in the clockwise and forms two coils. A group of coils collectively known as gene. నిజమే కదా? So, each and every 7 hours, for 7 hours, not more, but age factor is required, the limits of the age, DNA, two helicals. This helical forms to new helical. ఏమో అన్నాను. In how many hours? In 7 hours, each helical of DNA able to form synthesize to new helical, and it is coiling in clockwise. And this helical also forms to new helical and coiling in clockwise. Means two helicals. It is synthesizing like this. ఏం కావాలి? నువ్వు డిఎస్పి కావాలి. తలకాయ నలుసుకుంటావా? ఏమంటున్నా నేను? ఈచ్ హెలికల్ ఆఫ్ డిఎన్ఏ they forms to new helical in each and every 7 hours. Means the helical of the DNA duplicating, the helical of DNA replicating, the helical of DNA doubling. In each and every 7 hours, the helical of DNA have to synthesize to new helical. ఇది అర్థమవుతుందా? For each and every 7 hours, what is the character? Able to synthesize to new helical from each and every helical of mother DNA. Then after, during the time of replication or the duplication or the doubling of DNA, the new helical can be formed in the form of pieces. Then after, these pieces can be stitched by an enzyme. These pieces can be stitched by an enzyme. What the enzyme? Later we go to discuss. Now itself, what I can say, during the time of replication, what I can say, the new helical can be formed in the form of pieces. Then each and every piece by stitched by an enzyme is known as a ligase. What is an enzyme is required for the stitching of the DNA fragments during the time of replication. What concern replication? The synthesis of the new helical in each and every 7 hours from the mother DNA, they are able to synthesize to new helical, and new helical can be formed in the form of fragments at the pieces. Then pieces can be stitched by an enzyme is known as ligase. The method of the stitching is known as ligation. What is ligation? It is a method. In ligation method, they can be stitched to fragments into helical. కొంచెం అర్థమవుతుంది. ఇది మీకు అర్థమైంది? మళ్ళీ చెప్పాలి. Please, mallok sir. What I can say is 7 hours time is required. From common man to Superman. DNA have the 2 helicals. This helical can be synthesized to new DNA helical, but not in the form of helical. It can be formed in the form of pieces. It is the world. So that we consider the genetic material. Why consider DNA as the genetic material means during the time of protein synthesis, they are able to synthesize to mRNA, then after they are able to synthesize for this doubling. This type of the character nowhere we can see except DNA in entire organelles of the cell. So, for each and every 7 hours, what concern here, the helical of DNA able to synthesize to new helical, but in the form of pieces. Then after the completion of the pieces formation, the pieces can be stitched by an enzyme. What the enzyme is? Ligase. Then ligase can be stitched to fragments in a method. Method is known as ligation. What is the method means? You see, A, T, C, G, G, C, T, A, T, C, G. For example, you take like this. Then bonds with phosphate. It is a piece. What is this one? Piece of DNA. It is also piece of DNA. It is a piece. It is a piece. If you are applying ligase, what can be done? What can we do? Means ligase able to form phosphodiester bonds in between sugars. ఇప్పుడు కొత్తది అయిపోయింది. They can be stitched by an enzyme is known as ligase in the form of DNA new helical. ఏమో రెండు వేరు పీసులు కదా. ఇప్పుడు ఏమైంది? By applying of an enzyme ligase, what can be done means they can be do the formation of the bonds in between sugars. ఇట్లా ఇప్పుడు ఈ రెండు షుగర్ల మధ్యన ఉన్న ఫాస్ఫేట్ బంధం ఇక్కడ కూడా ఏర్పడుతుంది. కాబట్టి దే ఫార్మ్స్ టు హెలికల్. ఇది మీకు అర్థమవుతుందా? కాబట్టి నేనేమంటున్నాను అంటే ఈచ్ అండ్ ఎవ్రీ 7 hours the helical of the DNA forms to new helical. Then old and new collectively forms to DNA. And here also it is old and forms to new. Collectively forms to DNA. Means how many mother DNAs here? One. Synthesized to how many DNAs? Yes, it is nothing but replication. Also known as duplication. Means DNA synthesized to DNA. DNA synthesized to DNA. DNA synthesized to DNA. 2 DNA. 2 అన్న DNA అన్న నో ఇబ్బంది ఏం లేదు. బట్ వాట్ ఇస్ ద కండిషన్? వాట్ ఇస్ ద రిప్లికేషన్? సింథసిస్ ఆఫ్ DNA from DNA known as replication, also known as duplication, also known as autocatalysis, also known as Xerox. A Xerox of DNA. What is replication means? Doubling of DNA. Then this process can be controlled, catalyzed by an enzyme is known as the DNA polymerase 1. What is an enzyme? DNA polymerase 1. Polymerase. The name of the enzyme is DNA polymerase 1. It is enzyme. Under presence of this enzyme, only the synthesis of DNA can be formed from old DNA. The synthesis of DNA from DNA catalyzed by an enzyme. What the enzyme? DNA polymerase 1. Means DNA polymerase 2 is also there. But here the involving an enzyme is DNA polymerase 1. Under controlling of DNA polymerase 1, what we concern, the synthesis of DNA from DNA is takes place. For complete to DNA synthesis, the required time is, participated enzyme is, how many new DNAs are formed? 2. Is it clearer? During the time of cell division. During the time of cell division. During the time of cell division. Cell division. During the time of cell division. During the time of cell division. DNA undergoes for replication. Undergoes for replication. DNA undergoes for replication. You make the side heading to replication. Replication అంటే ఏంటో side heading పెట్టండి. Synthesis of DNA from DNA. Synthesis of DNA from DNA. Synthesis of DNA from DNA known as replication. Replication. Synthesis of DNA from DNA is known as replication. It's complete in 7 hours. 7 hours. It's complete in 7 hours. DNA synthesis completes in 7 hours. Catalyzed by DNA polymerase 1. Catalyzed by DNA polymerase 1. Catalyzed by DNA polymerase 1. Each replication it synthesize each replication it's synthesize 2 daughter DNAs. 2 daughter DNAs. In each daughter DNA, in 2 daughter DNAs. In each daughter DNA, half of the helical, conserving, half of the helical conserving, CONSER. Conserving means preserving. Half of the helical conserving from mother DNA. అవును కదా? Half of the helical conserving from mother DNA. And the remaining helical is new. Remaining helical is new. Early time of replication. Early, EARLY, early time of replication. Early time of replication. Daughter DNA synthesized in the form of fragments. Synthesized in the form of fragments. Synthesized in the form of fragments. These were stitched by an enzyme. These were stitched by an enzyme known as ligase. Ligase. In the method of ligation. In the method of ligation. In the method of ligation. Ligation. Next point. Replication of DNA. Replication of DNA takes place in the replication of DNA takes place in semi-conservative method. Semi-conservation means half of the helical of the mother helical can be conserving in new DNA. Semi-conservative method. Semi, not total, not complete. Semi means half of the helical conserving from mother DNA. In this daughter DNA also the half of the helical conserving from mother. Means semi-conservation. The half of the helical belongs to mother. The remaining helical is new can be formed. Method of stitching in between DNA fragments. Method of stitching in between DNA fragments. Method of stitching in between DNA fragments known as ligation. Method of stitching. Method of stitching in DNA fragments known as ligation. Ivala point. Ligase ఇచ్చాను. ఇచ్చినానా? రైట్. ఇది ఒక్కటే కొంచెం మీరు ఇది ప్యూర్ సైన్స్ లాగా ఉంటుంది. ఇది దీన్ని ఇంకా నేను ప్రిసైజ్ చేసి అంటే మీకు కావాల్సింది ఇస్తున్నాను. ప్యూర్ లో కూడా సెమీ ప్యూర్ అల్టిమేట్ గా దీన్ని అప్లికబుల్ లోకి వెళ్తాం. దీన్ని మళ్ళీ డెప్త్ అది ఇదని అనుకోవద్దు. ఇది డెప్త్ కాదు కానీ ఫండమెంటల్ గా కూడా కాదు. సెమీ దీన్ని యాజ్ ఇట్ ఈజ్ గా చదువుకోవాలి. ఇది ఎక్కడ పనికి వస్తుంది? మనకు మూడు స్టేజ్లో పనికి వస్తుంది. ఫస్ట్ ప్రిలిమినరీ, మెయిన్స్ ఉంటే ఇంటర్వ్యూలో కూడా పనికి వస్తుంది. లేదు కాబట్టి రెండింటితో సరిపెట్టుకో. ఏపీ వాళ్ళకి రాస్తే ఇంటర్వ్యూ కూడా ఉంది కాబట్టి అక్కడ కూడా రాయొచ్చుగా. మనం 15% ఎంతో ఇస్తారు కదా, 10% 5% ఇస్తారు. దాన్ని కూడా అప్పీర్ అవ్వండి. అది ముందు వదిలేస్తున్నాను. దెన్ వాట్ ఐ కెన్ సే, కొంచెం బోర్ అనిపిస్తుందా? బ్రేక్ ఎప్పుడు కానీ ఎన్ని గంటలకు వచ్చినాడు? మనం 7 కదా, 1 అవరే అవుతుంది. 9 అవుతుందా? సరే మళ్ళీ 9 స్టార్ట్ చేద్దామా? ఏం లేదండి, తీసుకుందాం బ్రేక్. జూన్ 9th కదా ప్రిలిమినరీ. దాని తర్వాత 4 months ఏమో దొరుకుతుంది. మీకు కావాలి తక్కువ టైమే. ఈ వారంలో ఒక 2 full days పెడతా. కొంచెం ట్యూనింగ్ అయిన తర్వాత మీరు నాకు నేను మీకు బాగా తెలిసిన తర్వాత ఒక 2 full days పెడతాను. 2 chapters లేచిపోతాయి, లెక్కలో కూడా రావు. అట్లా ఒక 10 days పెడితే ఫినిష్. మీరెవరో నేనెవరో సంబంధమే లేదు. మీకు కావాల్సింది మీకు చెప్పేస్తే మీరు చదువు. ఎప్పుడైనా ఆఫీసర్ అయితే అది వేరే విషయం.